All right, and in Chapter 5, what we're going to do is we're going to start building. Chapter 5 involves building large molecules. And the large molecules are built on the same principles of covalent bonds joining together atoms as small molecules, and then also built on the non-covalent interactions that occur between small molecules and that also occur between large molecules and the surrounding water environment and other chemical environments. In addition, so we have, we'll just write up here, large molecules they are joined together by covalent bonds. Atoms joined together by covalent bonds, so covalent bonds are a big part of it. For example, proteins are chains of amino acids, and so we get amino acids in our diet, like described in the very first video lecture, and then they get joined by covalent bonds to make long chains of proteins. So large molecules are joined by covalent bonds. Then in addition, there are non-covalent interactions. And they are between the molecule and the surrounding water. Or, in addition, other surrounding molecules. So non-covalent binding between, say, hemoglobin and oxygen, like that. And then also non-covalent bonding within the molecule, within the large molecule. So the molecule is big and there are interactions within it. So for example, a protein is a long chain of amino acids, and that's the chain of amino acids joined together, and the amino acid chain is long and likely folds back on itself, and there can be non-covalent interactions between this amino acid and one that's very distant along the length of the chain, but close in the neighborhood of the folded protein. And we'll get into that because that's part of Chapter 5. So the next thing to say about Chapter 5 is it's built on this, and then let's see if we can get to a little more space. It's built on this, and then there are some sort of types of macromolecules. And I don't like Chapter 5 so much because the author just kind of lists it well and describes them in detail, yet some of the molecules we won't study until late in the semester. So I wish that we could introduce them as we go. And so one macromolecule is polysaccharides, that's talked about. And we're not going to talk about polysaccharides until we talk about metabolism, because polysaccharides are chains of sugars, like glucose chained together to make glucose polysaccharide or polymer, and that could be glycogen, so a way of storing sugars in our liver and fat cells. Or other cells, all cells could store it as glycogen as a ready source of energy. Or in plants, in the form of cellulose. And so lots of different sugars, chains of sugars, but we won't really focus on this until later in the course when we talk about metabolism. Then another thing is nucleic acids, which are DNA and RNA. And DNA is, probably know, A, G, C, T. It's long chains of letters of nucleotides and that are joined together as covalent bonds. And so lots of covalent bonds. Then talked about in Chapter 5. Then another macromolecule that you're less likely to be aware of are called phospholipids. And phospholipids make up the cell membrane. So every cell on our theme handout, a big 7 theme, is every cell includes a cell membrane. So they make the cell membrane. And that's dealt with a lot in Chapter 7. So what we're going to do is we're actually going to learn about phospholipids now in Chapter 5. And we're assigned to read all this stuff. We're going to focus on phospholipids to tell some of the principles. Then we're going to actually go through Chapter 7, building the cell membrane. And once we've talked about the cell membrane surrounding a cell, we're going to go back to Chapter 5 and talk about proteins. And we're going to talk about a protein that's in the cell membrane. I've already mentioned it. It's aquaporins. That's the protein that was in the plant roots that let the water in the plant. So we're going to talk a lot about phospholipids, even though you're not that familiar with them. And then we jump ahead to Chapter 7, and then we're going to come back to Chapter 5 to talk about proteins, because we're going to be building a membrane, and membranes have proteins in it, and so we're going to come back to Chapter 5 and talk about protein macromolecules. And aquaporin is an example of a protein, like that. Okay, so we're in good shape. You can see that we're going to read through Chapter 5, but we're going to deal with it as we go. All right, so we start out with these figures. So simple molecules get joined together to make larger molecules, and there's the rules that we've learned about in the first couple chapters of building a molecule based on the principles of the atom. And, I guess I had already written it down. So those are the main molecules that are introduced in Chapter 5. And here's the one we're going to talk about. And this is a figure from Chapter 5, I think. And this is a phospholipid. And in our themes for the course, it says that all cells exist in a water environment. And then it says that all cells are surrounded by a membrane. Evolution is stories of patterns, and some pattern similarities and some differences. So here's a big time similarity. All cells, every kind, plants, animals, bacteria, yeast, all cells on our planet are surrounded by a cell membrane. And the cell membrane made up of phospholipids. So all your cells are surrounded by phospholipids, and it is a macromolecule. And that's about the size of it, so it's a relatively small macromolecule. Proteins are maybe 10 times this size. So if our cell was Boston, this thing would be the size of, well, not as big as a car, maybe the size of like a big bicycle or something like that. And these are atoms, and here's a nice drawing of it, and it shows you this. See this? That there are these two tails joined together by covalent bonds. And the tails are actually made up of fatty acids, the stuff that make the fats in our tissues. Long chains of fatty acids, and we already said that this is very water-fearing or hydrophobic. And shaded here, it shows that this is going to be pushed out of water if water is this white color on this page. These hydrophobic tails will be pushed out because they're all carbon-hydrogen bonds. Carbon-hydrogen has comparable electronegativity. These are all nonpolar covalent bonds, and they get pushed out of the water. Just to sketch it in, people will draw a phospholipid like this. This will be nice. Here's a phospholipid. And here's the membrane. The membrane is a phospholipid bilayer. Also, membranes are phospholipid bilayers. 2 layers and surrounding the cell. So all around the cell is a phospholipid bilayer. And so the central part of this core is very hydrophobic, water-fearing. And so out here is a hydrophilic water environment. In here is a hydrophilic water environment. And molecules have a hard time crossing this because it's a hydrophobic barrier. It's a very thin barrier, but it's hydrophobic, and things just can't get from one side to the other. And that's one of the big organizing principles of life, is that life is an organizing factor, and a big factor here is that the cell is like a bag that you can organize. So the cell can be floating in water. It can have highly organized structures and chemistry inside of it that are different than the outside. And so we want to zoom in and look at this. Remember this idea that covalent bonds don't stretch or bend, but they can twist. And so these tails are pretty floppy, but because they're hydrophobic, they get packed together pretty tightly in this phospholipid bilayer because there's that sort of core hydrophobic region. And then I already circled this. This part up here is called the head. So we've got something that has a head and a tail. Hydrophilic head, because look at the charges up here, very hydrophilic. Would love to, would love to be dissolved in water. And so that's why the phospholipid bilayer is organized this way. It has an outer hydrophilic region, an inner hydrophilic region, and a hydrophobic core. So water on the inside is attracted to the membrane, water on the outside is attracted to the membrane, and the membrane's being held together by non-covalent interactions, weak interactions. And so the next thing to say is to say that, again, this is one molecule, atoms joined together by covalent bonds, and it's a lot bigger than water. You know, here's water. So we've taken the principles and we've scaled up to the level of the phospholipid. And here's a nice schematic of a phospholipid bilayer and showing it in some detail. And that's that. So these would be things that are covered in, we jump ahead to chapter 7 to learn about the membrane. And the membrane's composed of phospholipid bilayer, and then plus what these purple things are proteins. So a protein's going to be in the membrane if it has a function in the membrane, and one of the proteins that's in a membrane is this protein that's called aquaporin. Remember that when we say we have aquaporin in our cells, we have lots of it. There are thousands and thousands of copies of aquaporin, and what aquaporin looks like is it looks like a donut. It's floating in the membrane because it has a hydrophobic region on the outside of the donut, and it's floating there, and there's a channel, a hole in the donut that lets molecules in and out. And aquaporin lets water selectively in and out, and we're going to learn about that as our first example of a protein that we learn about in detail. So we're going to use that aquaporin example to learn about the cell membrane and about proteins in the cell membrane, and then we're going to use it to turn back to some of the chemistry of the proteins. So if you look at this, these are all membrane proteins, and aquaporin is a membrane protein, like that. Aquaporin spans the membrane. It's on both sides of the membrane, so it's called a transmembrane protein. So here's the phospholipid bilayer, like that. The outer surface of aquaporin is hydrophobic, and these regions I didn't mention before are hydrophilic. Both the outside and inside are hydrophilic, and then we have a hydrophilic core that allows water to go through. Here's a nice drawing of phospholipid bilayer, and it forms spontaneously. That means if you took some phospholipids and you dissolved them in water and shook the bottle, the structure would fly apart, just in the same way that oil flies into tiny droplets. When it flies apart, the phospholipids may sometimes form a micelle, so just a little severe with a hydrophobic core and hydrophilic heads. Or it might make this phospholipid bilayer that we're learning about in these chapters. And so that's what where we're going to stop with this one.